So we've talked about the process of transcription and translation, going from a segment on the DNA and copying that gene segment, copying those instructions into the form of a messenger RNA. That messenger RNA then moves out into the cytoplasm, meets up with ribosomes and transfer RNAs, which helps it build a polypeptide chain that is then folded into a protein. So that's a lot to go through and a lot of moving parts to make that protein. What if there's a mistake that is made? What happens then? So that's what we want to take a moment and talk about. And these mistakes are called mutations. Sometimes these mutations occur when a protein's being made, or it could be a mutation that happens when the DNA is copied. So this can happen during DNA replication again. And that's a little bit more severe if it happens during DNA replication because that means every copy of that DNA will have that change in it or that mistake. So every time the DNA is copied, that mistake will be copied over and over and over again. If it's just a mistake that's made during transcription or translation, then at least there's a chance that when that gene is made again or read again into a messenger RNA, the mistake won't happen. So usually when we talk about genetic disorders or genetic conditions, it's at the level of the DNA where the mistake has been made and it's been copied over that way. So there's a wide variety of what we call different types of mutations. So we'll talk about point mutation first. So point mutation refers to just one point or where one nitrogenous base is different. Sometimes we call these base pair substitutions. So we have the example here of a normal hemoglobin, and it's nice and round and has a little dimple in it. And then we have a sickle cell red blood cell. So the sickle cell red blood cell obviously has a very different shape than our other red blood cell on the left-hand side. The normal hemoglobin sequence, which this is just showing a small segment of it, has GAC-TCCT. The sickle cell hemoglobin sequence, once again, just a portion of it, but it has GAC-ACCT. So the T has been changed to an A. This is a mistake. This is a mutation that has occurred. It's a point mutation because it's just in one base. So it's a change in just one base that has occurred. But a change in just that one base, look at the results. All right, so going from the nice round red blood cell to the sickle cell shape because of just that one base change. So it can dramatically change how the protein that is produced, the hemoglobin that is produced from that sequence. Other times it may not be that devastating. All right. So it just depends upon the molecule. It depends upon the amino acid that's being put into that position, and if that amino acid serves the same purpose as the original one, where in the sequence the mutation appears. There's a wide variety of different reasons for why some of these mutations can be very harmful and other mutations don't seem to be so harmful. But this is a case where just one base change can have very severe consequences. Other times we can have a base pair substitution and we get what's called a silent mutation. So a silent mutation means there is a mutation, there is a mistake that has occurred, but we don't see any difference in the protein that is produced. We don't see any difference in the amino acid sequence. So we have here what would happen if the DNA sequence of CCG changed to CCA. So we have a base pair substitution that has happened. The G is being changed to an adenine. So something very similar to what we saw with the sickle cell before. But let's see what happens. So if we have CCG, so here's our C, we go to our G column, and then we want to kind of go down and find where CCG is. right? And I think I drew my line right across from it, making it a little hard to see there. So let's see. Nope, I apologize. I went too far. All right, let's back it up a little bit. All right, so C, here's our C. So C, C, G. So CCG right there is proline. All right, so let me write that down, PRO for proline. Now what if it changed to CCA? So CCA is right there. Sure enough, it's proline again. Right, so there's a lot of overlap that happens in this codons with our codons because there are far more codons than there are amino acids. There are only 20 amino acids, whereas there are 64 different codons. So there's a lot of overlap, as you can see, where we've got many codons that code for the same amino acid. So in that case, this was a silent mutation because even though there was a mistake that occurred, the same amino acid was put into the same position, and the protein should function as normal. Other mutations are insertions and deletions, and these can be very devastating if they occur at the beginning of our gene sequence. So this is where an insertion would be where we add an extra nucleotide, and a deletion would be where we take out a nucleotide, and that affects the whole what we call reading frame. So this is an example where I have a sentence, the cat ate the rat, and each word is made up of 3 letters. So THE is 3 letters. That makes up my one word. Cat, 3 letters, 1 word. So I have this rule that every word in my sentence has to have 3 letters. What happens if I delete the letter C though? So we have a deletion, but I have a rule that every word has to have 3 letters. So now I would have the, THE, that's still 3 letters making up my word. But now C is gone, so all I have is A and T. So I write down my A and my T, but I have to have a 3rd letter. So I grab the 3rd letter from the next word, and I place it there to make up my 2nd word. So my 2nd word is good now because I have 3 letters. But now I look at my 3rd word and I only have T and E. So now I have to grab the T from the 4th word and place it there next to it to make a 3-letter word. Now my 5th one, I only have an H and an E. So now I have to grab another letter from the word downstream. And now I've made a word. And then we don't worry about these last 2 at the end here. But now I go from this sentence of the cat ate the rat, which makes perfect sense, to a sentence that now says the Attitaterat, which makes absolutely no sense at all. And that's what happens when you have a deletion. Everything downstream is changed. So if we have a deletion that happens within our DNA strand, and we start to build our protein from it, we're going to have to change our codons because our codons have to have 3 bases. So that means just about every amino acid downstream of that mutation is going to change. And the same thing happens for an addition, right? You're just moving in the opposite direction though, where you have too many letters and you have to throw the extra base into the next codon. So let's do some examples here. So we have a DNA sequence of GTC, AGA, GAA, GTA, ACC. So this will be our normal one, and then we'll have a deletion down here that we'll compare it to. So you can see that the A is missing in that 2nd codon. So you can stop the video and give yourself a moment to create the messenger RNA sequence from that DNA sequence and then find the codons that correspond to it within the chart below. So our messenger RNA sequence would be CAG, right? G is across from C. If we have A on our messenger RNA, we have thymine across from it on our DNA, G and C. Since we're making RNA, we would have uracil across from our adenine. We wouldn't have any thymines at all. And we're breaking it up so every 3 bases is a codon. Then you look up CAG in the chart here, and you'll see that CAG is glutamine, UCU is serine, CUU is leucine, CAU is histidine, and UGG is tryptophan. So this is our normal sequence. Now let's do this again, but having a deletion and see what the effects are with this deletion. So you can stop the video here again, create your messenger RNA sequence and your amino acid sequence. Once again, you need 3 bases to make up a codon, so you want to grab a base downstream to complete your second codon. All right, so our first codon is fine. So it would be CAG. Our next one will start with G, so we would have C across from it. A would be across from U, and now you need to take the G from the third codon to complete your second one. So G would be across from C. Then your next one would be A across from U's, and then you take the next base from the next codon, G across from C, and so forth, and so on, right? Okay, and then the last 2 I didn't include because they wouldn't make a codon. All right. So then we've got the amino acid sequence. If you look it up in the table, CAG is for glutamine. Now instead of serine, we have leucine. Instead of leucine, we have phenylalanine. Instead of histidine, we have isoleucine. So you see that the amino acid sequence is completely different after the glutamine. And that's what happens with a deletion. You're changing everything else pretty much downstream of it. And most likely this protein is not going to work at all because everything's different. It's probably not going to function at all because it's such a drastic change to that sequence. So practice these a few more times. All right, make sure that you can work out a deletion, create a messenger RNA strand, and be able to create an amino acid sequence if you're given a chart like this.